this afternoon, Christie Sayes from Baylor University is going to give us an overview of that interesting topic. So, Christie, do you want to use the microphone or?
Yes. There's a microphone at the left. I can use that.
Okay, perfect.
Thank you.
Thank you so much for the kind introduction, and to Norb for inviting me to this wonderful workshop. I'm glad to know that this particular center exists, and I hope to engage with Chris for as long as you guys will have me. I think it's a great public-private partnership that you guys have going here. And Rob is absolutely an authority in this particular area. He follows the literature and is actively pursuing research himself. So I definitely endorse everything that he has said previously, and I invite everybody to follow his publications in this space. He's very knowledgeable, and he's traveling around a lot to attend many microplastics workshops that are occurring around the world. So I was brought into this through Rob and through American Chemistry Council to think about microplastic particle reference materials for the testing that has been going on and will continue and should go on over the next few years and decade. I come from I'm a trained nanochemist, so I come from nanoparticle world, in which in the early days we struggled as a community to think about what those reference materials might be. The same type of needs that we needed 20 years ago for nanoparticles are the same types of needs that we need in reference materials today for the microplastics, and of course any other type of emerging solid state material that we're faced with today. We need positive control materials, those materials that can elicit a positive response. And of course, we need negative control materials as well. And it's also very important, especially as we learned from the immunologist earlier today, the need to characterize the vehicle for which we are testing as well to look for those particular effects. I'll start out with my research group. We are an advanced materials environmental health research group. We're made of postdocs, graduate students, and undergraduates. We aim to take a deeper dive beyond cellular viability after exposure, specifically to complex mixtures. It's why we have to build these fairly large research teams within my group. Most teams are comprised of 4 individuals, and we look at complex mixtures and build isobolograms. So if you're not so familiar, this comes from the pharmacology type of world for those of you who are familiar with isobologram analysis. But this is where you understand some toxicological effect of one chemical versus the toxicological effect of another chemical, and then you do complex matrix. Complex matrix are a binary exposure of different ratios of concentrations of those 2 different chemicals and put them together and figure out if that combinatorial effect is synergistic, additive, or antagonistic compared to the exposure of the individual chemicals. This is how we're working with microplastics right now. Microplastics in and of themselves are mixtures, but as I mentioned in my question to Rob, we also think of microplastics as one part of a complex exposure that might be in drinking water or in an aerosol exposure. So in drinking water contaminants, we look at the microplastic, their effects with and without binary exposure with disinfection byproducts or pesticides or metal ions or nanoparticles. And with air pollution, we look at liquid droplets and some other solid particles in combination as well. We also put an emphasis on looking at chemical transformations while they're in the environmental matrices. And so it's very important for us to understand how the environment might change these particles. And all of these are considerations that roll up into reference materials. The microplastics, this is as a take-home message, the microplastic particles, they can be simulated in the laboratory, and they can be produced and characterized. And I'm a firm believer of that. We have some methods that we've developed in the laboratory, and we're working with other partners to be able to improve upon those methods and come up with what's called a minimal base set Those of you that come from the nano world, you remember there was a minimal base set of particle characterization, as well as a minimal base set of toxicological endpoints that were needed at the very beginning when we started to produce the plethora of nanotox papers in the literature. The same type of thing will have to be needed for the microplastic literature as well, as Norb and Rob and all of us have listened before. But we also have to keep in mind that microplastics are not the only thing in the system and what the exposure might be. So we have to also think about, when we're thinking of the reference materials, what are particle-to-particle interactions and then chemical-to-particle interactions as well. Okay, so I'm going to end this talk with some research needs. I'm going to go through a little bit of preliminary data, but I'm actually going to save a lot of preliminary data for Beauchamp to talk about his studies after I speak. We already know that the plastic debris is everywhere. It's ubiquitous. It's highly variable though. So it's mixtures upon mixtures embedded in a enigma of mixtures. The microplastic itself, if you have a population of microplastics that you harvested from the environment in some way, there is multiple sizes, shapes, colors, polymer types, and binder types that are in that particular population. So that's the first thing to recognize when you're making reference materials is, do we make mixtures of the microplastics, or do we make homogeneous chemical composition microplastics? And right now, the people that are working in this space, including NIST and ACC, researchers from University of Hawaii are working on this as well, but both are on the table. You make homogeneous and chemical composition microplastics in different vials, and then you make ratios and mixtures of those different individual types of homogeneous chemical composition. They're heterogeneous in size and shape, homogeneous in composition, but you take aliquots from each one of those and mix them together, and that becomes a mixture of microplastics that you would see, that you would collect in some sort of waterway or in the beaches of the North Shore of Big Hawaii, the Big Island. We have to keep in mind that these things have modified surfaces, large size ranges, chemical additives or contaminants, the fibers and the particles both exist. And the most challenging one is, how do we accelerate the aging or the weathering of the microplastics you make in the laboratory so that they simulate environmental conditions? This is a different type of literature review than what Rob put up here. This is of the last few years that are looking at, not as time goes by, but simply the number of publications in these 4 different areas that my grad student and I are monitoring really closely. If you asked me just a few months ago, a year ago, the blue bars, the review articles, would have been higher in number of papers than the actual research papers in that particular space. Luckily, in the last year, we have research papers that have boomed, that made a huge boom into the literature. Now, of course, we need to look at those papers and understand how their experimental designs and, of course, their conclusions were drawn. There may be some room in improvement on those published papers, but I am happy to see that the number of research papers are far outnumbering the number of review articles and perspective pieces that are out there, which to me is a good sign for the science. But there is a understanding, because these studies that are put out there under toxicity, absorption, detection, identification, characterization, and weathering and aging they lack descriptive they are descriptive studies, and they lack a mechanistic analysis. The other point to mention too, Rob's literature review focused on a PubMed literature review. This is from Web of Science that includes those cited in PubMed, but also includes those chemistry and engineering papers that aren't necessarily cited in PubMed library, but they are included in Web of Science. I don't need to tell you that the micro and nanoplastics are widely distributed in the environment. There are multiple papers showing that they exist in freshwater, inland waters, for those limnologists out in the audience, marine ecosystems, marine waters, and of course, the aquatic birds, fish and birds. And then of course, in sea ice in Antarctica, the microplastics has been determined. But this is very important. that we know that these microplastics exist, and we've all seen pictures of beaches that are littered with microplastics, but attaining these real life samples and using them in mechanistic studies is not sustainable. So we do have to think about this and work with NIST to be able to put something together that is a reference material that's not just the polymer beads, but are actually fragmented degraded particles. And this stopped advancing. Oh, here it is. Oh, it's because I have micrographs. Okay. All right, so in the production of microplastics, we use a variety of different types of grinding machines. Blenders, of course, are the first line of grinding that we make in the laboratory, and these are kitchen commercial available blenders that can use dry ice and liquid nitrogen in their vessel. The vessels themselves are made of stainless steel, no plastic parts on any of these blenders that we have in the laboratory. You do need the liquid nitrogen in order to get fine materials. The top picture here is we're in particles in the fine size range, and the bottom we're in the ultra fine range here. We're less than 1 micrometer in a lot of these particles, and these particles are bigger than 10 micrometers in size. The large populations are fairly large, jagged, irregularly shaped materials. And then we have these very small populations as well. The trick is to size separate these particular types of materials whenever you produce them in grinders, in grain mills, and the third are ball mills with planetary motion. You can get very small size populations from these, but you have to be able to separate them. And right now, the sieves that are used in research labs like mine and around the country are not small enough to be able to separate out the quote nano-sized particles from the ultra-fine size particles. So we will need to be able to develop additional separation techniques as we continue to create these particles from a top-down approach. The categorization that we're using is done through size and the size and the surface properties, oxidation and the carbonyl content, which I'll talk about data sets from each of these in a minute, trace metal impurities. A lot of the colored plastics will have some sort of metal doping in them. Sometimes it's pigmentary chemicals, organic chemicals, but sometimes they're metals that produce the different colors. What the volatile and semi-volatile content may be, and then crystallinity. Here are a variety of SEM pictures that Clancy has taken showing the different surface features. The surface features are a little bit different depending on what type of microplastic is here. These, I believe, are all different polystyrene materials, but they're from different sources. So some polystyrene that from cutlery, and then polystyrene that's from the lids of coffee cups, and different other types of polystyrene ropes we've used and ground. So they have different surface properties as you go on, and I have more images later. So right now in the library that we're using for our laboratory, we have these 7 different lab-generated microplastic samples that we're routinely using for read-across type of studies for the characterization, and then eventually collaborating with Bo Chen to take all these and test them, of course. We're size separating each one of these vials into each one of the microplastics in each one of these into 3 different size populations with our sieves, and then testing them separately on the different sizes and of course what those effects might be. Plastic coffee lids, tennis ball fibers. For the fiber ones, we're actually using like a zester, you know how you would make lemon zest or zest your orange for cooking. We're using those grates to be able to get the fibers off, and then we flash freeze them and grind them further. Ropes, polyurethane foam, and packing peanuts are some of the easier things to grind and get to an ultrafine powder without having to move past the blender. We can use the kitchen grade blender just to get all the way down very small ultrafine size particles without having to subsequently take it to the grain mill or then take it to the planetary mill after that. So the minimum base set for characterization that we're using for each one of these particular plastics And what my grad students and I are looking for whenever we're reviewing papers that are published in the literature are these 5 things over to the left-hand side: scanning electron microscopy for the surface properties. That's connected with the energy dispersive X-ray spectroscopy to get the surface composition. Then we're taking it to the brand new differential scanning calorimeter, DSC, to understand the crystallinity of the different microplastics. As you know from the nano literature, the more crystalline a material is, the more likely it is to induce some sort of toxicological response. I'm not standing here saying that that's the same generalization we can make for microplastics, but it was something that some people have noticed with other types of solid state materials. So that might be a thing to measure and to report on. TGA will help us determine the volatile content that's in the microplastics. And of course, the FTIR is turning out to be a really reliable place to be able to get the carbonyl index, which is and other functional groups, but the carbonyl index is that characterization that tells you how much oxidation that microplastic experienced, either in accelerated aging in the laboratory or if you harvested it from the environment. Additional advanced characterization include whether or not these microplastics have a propensity to form and grow a biofilm on their surface, the isothermal analysis that I alluded to before of whether the microplastics tend to adsorb or have some sort of absorbance of other chemicals that are in the water as well. And of course, toxicity effects, which we've started going into, but it's very difficult and we're just not ready to start doing some toxicology ourselves. What we are noticing after weathering, after artificial weathering, is we see a huge increase in the amount of cracks and crevices in the microplastic surfaces. We also see nanoparticles form. This is indeed the same composition. This little dot right here is the same composition as what the microplastic surface is behind it. We see a lot of flaking that occurs. So these flakes will flake off and they may be large in one dimension, but you turn it on the side and their thickness is very, very small. So they're like platelets there. And then we're seeing these crystalline surfaces either revealed or form, especially when we put them in the environmental chamber and we increase the heat. And the sun simulator lamp is on full blast to accelerate the aging process with the sun simulator lamp. And we see these phase changes occur. This is a list of the environmental stressors that we're working with. We start out with the pristine microplastics that we make in the laboratory or fibers. We've also done some of this work with the microbeads. We think about the different environmental compartments, but mostly we're in water right now. And the environmental stressors, we focus on UV radiation with the sun simulator lamp and thermal oxidation. And we've worked a little bit with salinity, of course, the mechanical agitation, but we haven't really touched biochemical degradation or chemical oxidation yet. Those are going to be our next steps. And then for transformation mechanisms, these are the endpoints we're looking for to see how the microplastics, how each of these components change, increase, or decrease after you put it through the accelerated weathering process. Here's an example of the difficulty in finding that carbonyl index. The carbonyl index, you use FTIR to find this particular peak. Sorry that these labels are shifted over, but if you just look at the arrows are indicating the 3 different wave numbers that you look for these carbonyl indices. 2 of them are double bonded COs and 1 of them is a single bonded CO. It measures both the stretching and the bending frequencies of each of these particular peaks, and together it'll give you an index. You can see after a very short, this is only the microplastics were in the environmental chamber for 2 weeks. We saw virtually no changes in the carbonyl index after the microplastics were exposed to each one of these particular things for only a 2-week duration. So when we're looking at the accelerated aging, we have to think long and hard about this and do a lot more studies to see how long we will have to keep the particles in accelerated aging weathering situation and see how that matches up with the weathering that is existing in real life microplastics coming from the beaches and other waterways and other sources. biofilms. So these are actually microplastics that were harvested, that were given to us from a laboratory on the Texas Gulf Coast that are collecting microplastic materials. And we are seeing these colony forming units grow around these particular microplastic materials. So these they're labeled as control samples here because we didn't add any antibiotics or any bacteria. These plates were not streaked with bacteria beforehand. It was just the agar and these discs that were incubated with the microplastics and then plated on the agar, and these colony forming units popped up for all of these different studies that we had, the different plates that we had here. And isothermal analysis, these are some of the graphs that we produce when we change the pH of the microplastics in solution. We measure the paraben as a target analyte. It's a standard that you can buy from Sigma Aldrich, and that's what we put in water. And then we're looking for the paraben concentration using HPLC at different pHs of the solution to look for the removal efficacy. And then we also looked at contact time for how long are those microplastics stirring in the vial with the paraben in water over contact time. And we see over time it increased the amount of paraben associated with the microplastics increases over time. All right, so I don't know how much time I'm doing, but I'll quickly. Right, okay. It's, we see here, just keep going. Okay, so I'm going to switch a little bit to particle interactions and particle interactions and particle chemical interactions. And there's a need, in my opinion, to define these chemical mechanisms, which will also support the reference material development. Most of the studies reported in the literature are limited to single particle species at a time. But we do know that there's influence of other things in the environment, such as lipids or metals or other polymers. There's other things in the system, in the systems that will, just like with engineered nanomaterials, absorb and interact with the microplastic systems. And as we also know, as the surface properties or the surface characteristics increase and are different as the particle size decreases over time. So to illustrate that, there's this cartoon that we created for microplastic chemical interactions in the aqueous environment. Here is just a line that is separating the ocean from the air above it. So I'm such a great artist. I think that conveys the sky versus the water. This is natural organic matter, as we all know, that is in water naturally, no matter where you are on Earth. And here are some microplastics that are falling out of the sky from some unknown point source. And when they come into the water, they do one of two things almost immediately. They can start degrading due to their interaction with sunlight. And if this is salt, if this is ocean water, then salinity there. They also almost immediately complex with natural organic matter. And then this complexation, it's immediately heterogeneous. And it can include the primary microplastics as well as partially degraded microplastics. At the same time, we're also releasing other material substances into the environment, things like disinfection byproducts or pesticides, metal ions, and of course, engineered nanomaterials like iron oxides. All of these things are also being released into the water at the same time. It of itself is a heteroaggregation. But what we know happens to this type of aggregation, they release ions, especially if it's a metallic substance. It releases metal ions, or if it's something like a pesticide or disinfection byproduct, it goes through other transformation processes as well. Each one, on both sides of this, once the sun enters the environment here, you have the photoactivation, and dissolved oxygen also exists in this waterway. So you have photooxidation that can continue to degrade and change the surface of the plastics. You have dissolved oxygen, which can also contribute to some oxidation processes going on. And then you have this secondary complexation of the natural organic matter with other chemicals, other degradant products, and the microplastics itself. So all of this gives you a really strong headache whenever you start think about how can we do the weathering process for the reference materials. It gets very complicated really fast, but somewhere buried in all of this is probably an expert working group that could probably tease out what are some environmental processes that we ought to simulate and then be able to put reference materials through in some sort of standard method development. If you boil these down, we have degradation, complexation, primary complexation, heteroaggregation, release of cationic or anionic species, photoactivation, oxidation, and secondary complexation. We ran one study where we actually incubated a PAN, polyacrylonitrile with organic matter and silver nanoparticles together to be able to produce some studies. Here is the TEM image of those silver nanoparticles. That's what the darker spots are silver nanoparticles here in the middle. And this is what's presumed to be the natural organic matter and the PAN particles. The transmission electron microscopy confirms nanostructures in this particular complex here. And we collected FTIR to show the carbonyl groups and the plastics are present. Aromatic ring groups are present as well. So presumably this is the PAN. This is representing the silver nanoparticle. And we are hypothesizing this should be more of a dotted line, that this is the association of the silver, either in the ionic form or the nanoparticle form with the nitrile group here. We've also incubated the silver nanoparticles with PET and with PS to be able to form other types of complexation as well, but we only tested the first one. The PAN in microplastics in the pH itself has a nominal size of around 80 micrometers. But then you change the pH, doesn't really change. You put some salinity in it, some salt in it, it decreased. You irradiate it with the solar simulator lamp, went really small. And then as soon as you complexed the PAN with the natural organic matter and the silver, the particles got really large, presumably because of the natural organic matter. As shown here, the PAN with the natural organic matter by itself, very large particles. But with the PAN and the silver complex by itself, we're still around the same nominal size population as the PAN itself whenever it is in environmentally relevant pHs. These are just some more, I just want to throw this in here just to show you more, I put this in the wrong place, but this is more micrographs of the surface features of polystyrene, which doesn't have to do with what I'm saying right now, but just so that you see more surface analysis of the microplastics here. And when we characterize these microplastics, we see if you increase the magnification on each of these images with lowest magnification here and then highest magnification down to here, not only are we seeing the change, the heterogeneous nature of these surfaces over the different magnifications, but we start to see these really cool little pit formations here that are really interesting sites for which maybe other things can hide inside of. And of course, again, you start to see when you do EDAX on these smaller fragments here, they're the exact same chemical composition as the background is, telling you that these are the polymer samples. They're just in a separate nano-sized type of size compared to the background microplastic that is larger, closer to the 50 micrometers in size. Okay, and I will now, I'm gonna not talk about our human gut co-culture model, but I wanted Bo-Chen to know that we're also working with a gut model as well. But with our human gut cells, we did both an E. coli study with those PAN silver aggregates and looked at the growth, and that's this green line here. We saw in lower concentrations and the higher concentration, we saw inhibition of the growth of the E. coli growth after exposure to the pan-silver complex. It was most detrimental in light. This is particles were incubated in light, whereas the microparticles were kept in the dark. We still saw the less bacteria growth in this pan-silver complex than we did pan by itself or silver by itself. But we're seeing a decrease in both the growth of the E. coli cells as well as our human gut cell model as well. Significant decrease in viability of our gut cell model when the pan silver complex incubated in light and exposed to our gut cell model. So the pan by itself is decreasing viability, but to a lesser extent. We're thinking that there's something else going on here that needs further analysis and definitely pushes forth the need for us to think of these as complex mixtures. Okay, my recommendations for future needs definitely include characterization. We need to understand the same things that the WHO was recommending before, that Rob already mentioned. We need to understand pathways into the environment, point sources, extrinsic versus intrinsic properties of the different microplastic samples themselves. I firmly believe that the properties will dictate the effects. I think of this as very much similar to a nanotoxicology story. And I definitely think that we need to do as much characterization as we can to figure out what are those drivers. And we also need to understand the decomposition or degradation products that are coming from the microplastics over time. We need to have controls. We need to understand positive controls and negative controls. We need to have controls in the methods development, definitely a need for the reference materials. For human health, we need to understand route of exposure, susceptibilities, probably not any unique human diseases, but we may see some toxicity dependent on the particle size and the mode of action. And then in the environment, we can't forget about the environmental impacts that might exist as well for adverse environmental health endpoints that we need to have control of and understand as well. So with that, I will stop and thank you for the opportunity to talk today.
Wow, thanks for a fascinating presentation. Talk about complexity. Anybody have questions in the audience? Or anybody online with anything? While he's looking,
yeah.
We were just having a little sidebar actually about this, and we were just commenting about how complicated all this is.
Yeah.
And then, of course, we saw you present earlier that there's been a lot of papers published recently on this.
Yes.
Can we trust anything from any of these papers?
Well, the fact that Norb just even mentioned that some of the papers that are looking for immunotox endpoints maybe aren't using the right methods to measure those immunotox endpoints. I don't know if we can trust them or not. I mean, to me, this is the exact same story that happened 20 years ago with the nanotox literature as well. There was including myself, there was a bunch of people that were looking for cytotoxicity in maybe one particular cytokine and put out a paper because the literature was craving it and associate editors wanted those types of papers. So I think it'll probably even out as time goes on, and we just need to be more diligent in the peer review space and call out studies that are doing overdose concentrations for sure, and maybe aren't using immune cells when they're measuring immune endpoints. That's my biggest pet peeve with that. So we need to make sure we have immune cells in the system if they're going to go in that direction. Sounds like a good idea. But I think it'll even out. I personally still think it's good that we're having more research papers now than having people's opinions put out there, because for the longest time, it was just review articles and I didn't really understand what the authors of those papers were reviewing because there was no literature to review. So it turned into a lot of postulating from other solid state chemistries into this particular solid state chemistry, and some things were just not translatable. So I'm glad we have the research papers now so that we can write review articles that maybe are a little bit more informed.
So we do have one question here online. It says, it is excellent that you are using commercial products as a starting source of microplastics generation.
Yeah.
What is the smallest reproducible size achievable with your techniques? Also, are certain products more difficult than others to size reduce?
Absolutely. So I would say consistently, if someone asked us for some lab generated microplastic samples, we can provide them with 500 micrometer sized homogeneous size population. That's probably the smallest that I would feel comfortable sending, like a 50 milliliter centrifugation tube full of 500 micrometer sized particles. That would be the smallest, I would say. That we do produce particles that are smaller than that, but not in enough quantity to be able to share and be able to do any sort of study with.
So Christie, I have another question, and that is, see you here. So, you know, clearly one of the biggest obstacles in this area has been having reference standards so that one can compare across laboratories. I wonder if you could tell us a little bit about kind of the larger picture in terms of what are the efforts on a larger scale to bring folks like yourself together to actually produce enough of the same materials that they can be used by laboratories around the world? And is there such an effort going on through the ACC or some other organization?
Yes, it's NIST and JRC have definitely partnered together to figure out who might be the producers of these reference materials and where might they be stored, what repository might they be stored. That will come along when you purchase the reference materials. So those discussions are underway. I have a small investment in grain mills and blenders and ball mills in my laboratory, but it's nothing compared to the rooms that NIST has filled up with some of these things. And their grain mills are huge, like industrial sized grain mills that they've purchased and that they're working with. There already are, I don't know if anybody has visited the NIST reference materials site recently, but there are so many reference materials that NIST already has. So the idea is that certainly the polystyrene beads that they sell and they characterize and they source will automatically make the list for microplastic reference materials. That will already be there. These real life top-down jagged edged fragments, that's what the community is trying to figure out. Which one will they be? Will we just buy a whole bunch of plastic cutlery from Amazon and call it a day? Or will it be something else? They have standard reference materials on the list for very pure things like some rubber and even rubber that has been rubberized to be the solid state part of it. They have reference materials in other polymers, in monomers, and I don't really know what the form factor that those are, but that list of what they already are commercially available to buy from NIST already has, I think they identified maybe 20 to 50 different materials that might be able to make start the repository for that reference material. The question is then, do we want weathered reference materials? And that's the question that I'm interested in figuring out, because it's not just the method development for how we will produce these jagged edged microplastics, but what about the standard methods we're going to use to weather these things? Because if you weather them, what we're learning is that 2 weeks is not long enough to keep these things in an environmental chamber in order to see significant changes in the surface properties. So what is that? Is it 4 weeks? Is it 4 months? There are people that are actually putting microplastics on the ceilings of their buildings and waiting a year or two. This is what University of Hawaii has done. They have these vats of microplastics in kind of a slurry, and it's sitting on the top of their building, getting the Hawaii sun, and it's been up there for 2 years, naturally aging on normal time scale. So those are some questions that we need to understand. I think we should have maybe we don't need the reference materials for the weathered microplastics, but we certainly need maybe some SOPs around how to weather them.
Christie,
yeah.
As always, we enjoy your talk.
Thank you.
So regarding the question, how much we can trust the literature paper published, So just like I'm guilty about that. But when we're dealing with engineering nanoparticles,
Yeah,
10 years ago, we made all kind of mistakes.
absolutely. I did too. Yeah.
And 10 years ago, a friend from NIST and a few other people from Europe with popular paper, like artifact, we're dealing with toxicity.
Yes.
Dealing with engineering nanoparticles.
Yes.
We did the same thing. Just accept it. Probably we start submitting the proof. We have an article in Developmental and Technology, artifact dealing with microplastic and nanoplastic particles. So if anyone interested, I'm all dealing with ecotoxicity,
not human health.
Not human health. So for the characterization, I don't know if you would like. I'm just asking for comment. The molecular weight, polymer, because same polymer, the degradation rate can be different.
Absolutely.
The molecular weight.
Yes. And Dr. Andretti, Andy Andretti from NC State is looking at density and molecular weight as one of those maybe needs to be included in characterization in the minimal base set as well. So it's just that the density of the polymer is different than the plastic itself. And so that, it includes the binder and the plasticizer as well when you're talking about the microplastic, and we cannot fall in a trap of just thinking of it's the polymer that's inducing the toxicity, because a plastic is different than just the polymer. That would be the only thing I would say to that.
Absolutely.
Yeah.
That relates to the degradation rate.
Yes. Oh, it would relate to degradation rate. Absolutely. Yeah.
One more question regarding how you break down those particles.
Yeah.
You see the blenders, other.
Yeah. Grain mills. Ball mills.
We used to do that.
Yeah.
Biochar particles.
Yes. Yes.
When we do that, we take over persistent free radicals.
Yep.
Always there. I don't know that would be a concern when we produce a ultra fine microplastics.
Right.
Because you have free radicals there. Of course, radicals themselves will cause toxicity. In addition,
it's a good idea.
Small particles.
Yes. Yeah, we could probably run some ROS assays immediately after grinding them and seeing what we see there. That's a great idea.
I wonder if you could talk a little bit more about these reference materials.
Yes.
Specifically, what is your thinking as to how to validate the extent to which it mimics what's in the real world? In particular, it seems to me that if you look at these particles in the real world, it can differ in salt water and fresh water.
Yep.
There's an area where there's a lot of sunlight or not a lot of sunlight.
Right.
So after you go through all of this effort in terms of preparing reference material,
right,
how do you then validate
that
what you have comes close to mimicking what's in some area of the real world?
So for the reference materials, I'm never going to be somebody who would recommend that we characterize the toxicity of the reference materials, and then that would inform policy. What I would like to see is that people who are conducting toxicity studies are including at least one, if not two or three, whatever the community decides is the appropriate number, include a 1 to 3 reference materials in the study in addition to whatever real life microplastic they may be studying. And so you would make then the policies and moving forward based off of the effects of the real life particles. But then those of us who are into data mining and looking to be able to do read across studies, we have the reference materials there in that paper as well as any other paper. And then we can read across the papers of the reference materials to see where outliers might be against laboratories. But as far as the utility of the data collected, it would make more sense to me that we continue to find collaborators who are field researchers. They're harvesting the microplastics from an area of the world, and then we do mechanistic studies based off of the real life materials. That's where I would see it.